Hi, everyone. Welcome back to our masterclass series here at Active Concepts. My name is Morgan Drohan, a technical marketing professional here. You may recognize me possibly from season 1, episode 2 of our skin penetration episode. So this season is a little different. We are starting our season 2 and it's back to school. So everybody grab your pens and pencils and your notebooks because season 2 is really going to focus more on efficacy testing that we do in-house at Active Concepts. Season 1 was much more based on our biological functions and how that relates to the cosmetics industry and how those products are actually working on a more scientific level. And season 2 is going to kind of dig into what our scientists do here every day, whether it's to prove our ingredients are anti-inflammatory, whether they can increase wound healing, reduce cellular senescence, or even manipulate our skin's microbiome. And so today we're going to have Daniel Schill here with us, and it's a super special episode because not only is he our manager of clinical validation and toxicological assessment, he actually designed the assay that we are going to be talking about today, which is the Dermal Microbiome-Immunology assay. So Dan, I'll go ahead and let you introduce yourself.
Thanks, Morgan. Excited to be here. So briefly, I studied human physiology at James Madison University, then at the University of Georgia, lastly at the University of Maryland, where I gained a lot of research experience. And during my training, I was awarded a grant from the National Institutes of Health, performed a bunch of in vitro experiments, and was in charge of randomized control trials and other in vivo studies. So I entered the cosmetics industry with Active Concepts at the beginning of 2021. And while working here, I earned a certificate in cosmetic product safety assessments. So as a cosmetic toxicologist, I'm utilizing all of my skills to oversee our efficacy, safety, and toxicity testing at Active Concepts.
Awesome. Very impressive, and we're super lucky to have you on our masterclass today. So Dan, the assay we're talking about is called the Dermal Microbiome-Immunology assay. Quite a mouthful. But can you perhaps give us some background on what this assay is and what it tests for?
Absolutely. So this assay is based on background research on dendritic cells.
Stop. What are dendritic cells?
No worries. Dendritic cells are a small group of heterogeneous cells residing in the epidermis and dermis. Basically, they're known as the gatekeepers of the immune system as they recognize antigen-presenting cells that recognize when foreign pathogens enter our body.
Okay, so these cells are basically responsible for recognizing when something is wrong.
Yes. So when these dendritic cells are activated, they communicate with the rest of the immune system through paracrine signaling by releasing specific molecules which alter the function of nearby cells. So these dendritic cells play a critical role in maintaining homeostasis during bacterial infections and disruptions in the skin barrier.
Okay, so I see. So the knowledge of dendritic cells is really imperative to designing this assay. You mentioned them being the gatekeepers of the immune system, but how does that necessarily relate to the other components of our skin microbiome?
So the skin microbiota consists of bacteria, fungi, and viruses.
Gross.
Well, actually beneficial. So these components protect against invading pathogens, but also provide critical instructions for the innate and adaptive immune responses. So while beneficial microorganisms make up the majority of the skin microbiome, there are sometimes some alterations in the commensal pathogenic balance, which is associated with dermal inflammation.
Okay, so meaning an off balance of good and bad bacteria is a reason that our skin gets really inflamed or irritated.
Yeah, so perhaps not the only reason, but definitely a main one. And if we're able to restore balance to the microbiome community and reestablish this barrier by reducing harmful microorganisms, this may suppress cutaneous inflammation. So this is where the activity of the dendritic cells comes in.
Because the dendritic cells are the ones who are responsible for releasing the paracrine signaling molecules to kill those detrimental microorganisms.
Yeah, exactly. So in fewer words, this assay assesses a chosen active's ability to alter dendritic cell paracrine signaling to therefore restore the skin's microbiome.
So that's great. So that can really give a whole new perspective on ingredients as well. And kind of gives it the ability to basically rebrand ingredients that we've already launched. Interest in the skin's microbiome is still a really hot topic from a marketing and trends perspective, and nobody has really cracked the code on the skin microbiome yet. So, right now prioritizing one's health and skin immunity is continuing to skyrocket. So this is really a breakthrough for the personal care industry. So Dan, have you ever heard of inflammaging?
Yeah, so you mean one of the main reasons why one of the main issues I was trying to address while creating this assay?
Why, yes. So one of the leading causes of aging is actually stressed skin, technically referred to in the personal care industry as inflammaging, which is the chronic low-level inflammation with the degradation of collagen tissue and cells. And since this makes our skin more susceptible to outside environmental factors, that will increase the appearance of fine lines, wrinkles, and sagging skin. So maintaining our skin's homeostasis and immunity is extremely important. Therefore, if we can find a way to actually alter dendritic cells releasing those paracrine signaling molecules, we can actually prove enhanced skin balance through the manipulation of the microbiome. And I think I just heard influencers and brand developers taking notes because that is super important. So let's get into the assay. How do we set something like this up?
Yeah, so this is basically just another time kill test. So to set it up, we take monocytes and then differentiate them into dendritic cells according to the manufacturer's protocols. Then we cryopreserve them. And then to actually generate the conditioned media, which is used in the assay, we seed these dendritic cells into a plate with fibronectin on the bottom, which is just basically a basement membrane which allows them to attach. Then after 24 hours, the complete media is replaced with either complete media, with more complete media, which acts as spore control, or various concentrations of the active being tested.
Okay, so now that we have our solutions ready, can you explain the steps of a time kill test for people who might not know?
Sure, yeah. So the conditioned media samples are taken off and then they're frozen and then thawed. And then each of those testing conditions are added to individual containers that were inoculated with one of the 3 test organisms, so either S. aureus, B. aeruginosa, or S. epidermidis.
Okay, is there a significance as to why those 3 bacteria were chosen in this assay?
Yeah, absolutely. So S. aureus and P. aeruginosa are human opportunistic pathogens with antibiotic-resistant strains, while S. epidermidis is an element of normal skin microbiota. So it's non-pathogenic and supports healthy dermal barrier integrity. So the inoculum concentration for each organism is standardized using the McFarland turbidity standard, and then further diluted to yield a standardized concentration of microorganisms. So the amount of each inoculum added to each sample is more than 1% of the product's volume, so that way we don't alter the composition.
Okay, I'm following.
Yeah, a little complicated, but so next, basically serial dilutions from each container are performed to basically enumerate the surviving microorganisms using the plate count technique. And the activity of the test material inoculated is evaluated at specific time intervals, so 30 seconds, 1, 5, 10, and 30 minutes, and then results are read 2 days later after plating. Okay,
awesome. So when this assay is all said and done, how can we determine if it performed well? Like what kind of results are we looking for?
Yeah, so that's a great question, Morgan. So what we want to see is the prevention of the growth of the 2 detrimental organisms, the S. aureus or the P. aeruginosa, at every time point, and not so much with the commensal microbiota, which is the S. epidermidis. If that's the data we see, we can confirm that the ingredient tested enhances the ability of those signaling molecules from the dendritic cells to kill the detrimental organisms, while maintaining the growth of the commensal bacteria commonly found on the skin.
Which is a great clarification, because I feel like there's this common misconception that all bacteria is bad bacteria, when in reality, we don't want to kill all of the bacteria found on our skin. And the results of this assay can prove that while we're, killing the harmful ones, we're still preserving the good bacteria for our skin.
Exactly, yeah.
Awesome. So here's a question, Dan. Can any ingredient be tested on this assay?
Another great question. So sort of. So before running this assay, we perform extensive research and perform several pilot experiments on each of the potential ingredients. So what we do in that is we want to make sure our ingredients will activate dendritic cell paracrine signaling, but specifically in a way that we can harness the properties of those signaling molecules to restore balance within the skin microbiome and reestablish the skin barrier.
Okay, awesome. So that definitely clears up that maybe not all ingredients would perform well on this type of assay, and that would be able to claim to modulate our skin's microbiome. So why is this assay a breakthrough in the personal care industry? Immunocosmetics is a holistic approach to treating the skin as an ecosystem to work in symbiosis across all of its pillars. So while the skin microbiome was discovered a few years ago, being able to actually manipulate dendritic cell signaling is huge for the cosmetics industry, and it will really allow brands to have that next level advancement in the skin microbiome space. So Dan, thank you so much for taking time out of the lab to do our masterclass series with us. Again, this is Dan, our manager of clinical validation and toxicological assessment. I'm Morgan Drohan, a technical marketing professional, and we will see you guys next time for episode 2 of our masterclass series.